I'm just going to wait a few minutes for a few people to roll in, and then we'll get started. Sorry, going through the safety briefing. It's got some funny pictures in. Okay, so hopefully you all got the memo that this isn't in person today. Otherwise, you'll be kind of on your lonesome, unfortunately. All right, so what we have to do today are go through some kind of basic stuff, safety briefings and safety forms. Some of you have already emailed me about those, so I'll go through that in a second. And then we're going to go through, well, let me just make sure I've actually closed the assessment so you don't see what the answers are. And then we'll go through the actual lab for today. It shouldn't take too long, hoping on about an hour, give or take. But obviously, if anyone has any questions, please feel free to ask away. So this week's a bit of a special week, largely because my wife's out right now. I'm nominally supervising the kids, but they're actually pretty happy just doing their classes and stuff. And obviously tomorrow I'm going to be out for my immigration interview, which will be fun, I'm sure. So next week the labs start in earnest, I guess one could say, and the whole alternate sequence will begin. Right. So next week is lab 2. What is lab 2 actually? It's got to be. I should know this by now. I've taught this enough times. Just got a memory of a fish. Oh, there you go. Corn. Ha ha ha. So if you don't like your lab partner, make sure they go because they'll be canning lots of corn. No, honestly, it'll be lots of fun. Promise. So next week is the corn lab, where you're just going to be basically doing some Mendelian genetics and using chi-squared to figure out whether you got it right or not, or whether your assumptions are right or not, which is kind of more the point. And whoever's in that week will be attending. So there should be 12 of you, I think, and I'll get those lab sheets out, partners, out by the end of the weekend. So if you want to choose who your lab partner is, or even choose who your lab partner isn't, if you have a preference, you need to let me know by Friday, because I'll be putting that together over the weekend. And once it's out, it's done, right? I'm not going to mess around with it after that, because it just causes all kinds of problems if I do. So if you do have a preference, then please let me know, and I will make sure that preference is taken into account. So obviously next week, half of you will be coming into lab. I'm getting Zoom bombed by my daughter. You look like you're choking her, Sophie. Yeah, there you go. That's better. And my other, oh, they're my feet. You don't want to see those. Well, my feet. You've got esoteric tastes. I swear, when I was a kid, my dad called me a hobbit, uh, which I thought was rather rude at the time. Obviously, oh yes, didn't turn out to be entirely true because I'm quite tall. I'm not hobbit sized. Um, anyway, yeah. So uh, Luna the dog is busy sitting to attention. See how she sits like a human. She's very weird. I actually have some pictures. Let's see if I can find that now. Going on for a little bit of a tangent. Bum, bum, bum. I have a whole folder called cat and dog pics on my computer, which I think is. I know, Sophie. I just want to find you this picture because it's funny. I often post this pic when I say I'm writing exams. Come on, open up. There you go. I'll show you in just a second. Yeah, I know. There you go. That's Luna writing a Gen Bio 1 exam. You see? Yeah. She did a great job too. Everyone was very happy. Um, anyway, again, that's a bit of a tangent. Um, holy shit, bits. 12 pets. Wow, that is a lot. Cool working at a vet hospital. I wanted to be a vet when I was younger, in like 16 or thereabouts. Then I did work experience at a vet and discovered that I was astonishingly allergic and I couldn't breathe for like 3 or 4 weeks. So I didn't become a vet. Anyway, so obviously next week you'll be doing, half of you will be coming into lab to do the corn genetics lab. You need to share those data with your lab partner that you'll be collecting. So if you leave her alone, and all of you will need to complete the assignment on the course content page for that lab. So let's, yeah, there's Luna again. So the one for today is, this is obviously what we've got today, but if you go to lab 2, if you actually went there today, this is what I see. So this is like professor view, I guess. But you will see only the, get out of it, the lab handout, right? You will not see this assignment because it's only available after, I think it's like Sunday or Monday afternoon. Depends whenever I was preparing the labs. So you will see this assignment, appear magically on the 30th. Yeah, that's Sunday afternoon, essentially, right? If you're worried about it. And that's going to be the same for all of them. So someone thankfully pointed out that I got the due dates wrong. Weirdly, just for these 2 labs, I don't know why. But anyway, that's been fixed. So today's lab is due next Thursday. And obviously next week's lab will be the Thursday after that. And so that's generally the case for all of them. They're going to be due midnight Thursday after you take the exam. And it's just Thursday because that's because I have a lab on Thursday as well. So you essentially get an extra day, which may or may not help. I don't know. But anyway, just tell yourself that it's going to be Wednesday night and then you'll have an extra day in case you forget. And the only exception to that, I think, from memory, is the Eugene lab because it's a 2-week lab, kind of spread out over 2 weeks essentially. And that is due, December something or other. No, don't want that. I want this. Too many Word files open. Close. Yeah, so the Eugene lab is, December the 3rd. right? So that's the exception. And also the Excel spreadsheet lab, which you're going to be using your own flycross data to produce. That will be that, and the mini lab report deadlines are here and here, right? So you want to know when anything is due or what you're doing on a particular date. Go to the syllabus section on Blackboard and you'll find the schedule there for you. And that will tell you essentially whatever you're doing. I had a question about the lab book for the fly lab part. As long as the sheets of paper can't fall out of it on their own accord, doesn't really matter what it is. You don't have to buy a new one. You could use part of an older one or whatever. It's really not that important. As long as you're able to keep a record of what you're doing and the data you're collecting, and that record isn't going to go walkie somewhere or fall out in the corridor, you're totally fine. Let me just pop up I think I had a chat come up. All right, give me just a second. I'm going to have to go back to. Oh, good question, Mackenzie. So you each turn in your own assignment. So all the assignments are tied to your particular Blackboard logging. And so you will be doing your own. Now, obviously, you can, if you have questions, you can ask each other, but I do not want you to be simply copying what your lab partner does, because it's pretty obvious when you do that. And if you do that, then you risk getting 0 for that particular assignment. The same with the Excel spreadsheet and the lab reports. Obviously, you're going to be collaborating in terms of data analysis and the like, but you should be doing your own work on those assignments. Question from Gabriella about the person who's not in lab. So I'll be doing these Zoom sessions at the beginning of each lab. So the person that is not in lab at the time, I will be essentially giving everybody the same information in terms of what we're going to be doing, nature of the assignment, what you need to hand in, essential information. So the person that is not present should be able to get all of that either at that time or when I upload that to Blackboard, not Blackboard, YouTube after the lab has finished and it's had time to convert and so on. So that essentially the aim is that the person who's not present gets all of the background and information. They just have to wait for their lab partner to give them the data. So all they'll be missing out on is the actual doing, the physical motions of doing the lab. They should still get all of the data and do all of the analysis and assignments as their partner who is present does. Right. So, and that also hopefully should answer some of Elizabeth's question too, which is along the same lines. So the aim is that essentially everyone is getting all of the information and in terms of what the labs are and what you're going to be doing. And they do all of the analysis and you get all of the kind of analytical skills. But you'll only be getting half of the hands-on practical skills, I guess would be the best way of putting it. So that's the compromise for this semester, essentially. And yeah, so essentially that's what Elizabeth asked. Whoever's in lab, let's say next week, for example, you're going to be counting corn. You count your corns, you collect that data, you then email your lab partner those data after lab. And, if your lab partner who's not present has any questions, then you can go through that with them. So, pardon me, it's kind of like a virtual version of the two of you sitting together at the end of the lab going, what the hell does this mean? And figuring out how to fill out the lab handout, right? So that's essentially So Haley, there'll only be a Zoom for the first, however long it takes to go through the background information and the protocol and the assignments and stuff. Um, so that shouldn't take longer than an hour, hopefully. And anyone who isn't in lab will be able to join that. So I think I sent, well, I mean, you're all here, right? So you must have the Zoom link. But I'm not going to have the Zoom on for when I'm wandering around, helping people out, because you'd just be staring at a blank wall for another hour or hour and a half. And then once that initial part has finished, then I'll stop the Zoom and that will be posted up to YouTube afterwards. Also, we'll have this chat session open as well. Hang on a sec, I'll answer your question in a moment. So if you have any questions as we're going through the protocol, the assignment, whatever, you can ask them in real time. You don't have to wait until after the lab to talk to your lab partner. So you can actually ask me like, what do you mean this is how we're going to be doing the chi-squared? Can you go over that again? I can do that kind of in real time with you at the beginning of the lab. So hopefully you'll get as much of the experience as possible and the benefit of being in lab just without being there. Gabriella asked about video chatting privately with your partner. Of course, that's as if they were there, right? I have absolutely no issue with you doing that whatsoever. One presumes that the university Wi-Fi will cope with that. I hope so. I don't see any reason why not. But yeah, totally. I think that would be awesome. I can't help you with that because I am technologically illiterate. This is kind of figuring out how to use Zoom is about as good as it gets for me, to be honest. But yeah, if you want to do that personally, that's totally fine. If you want to do that after lab, if you have any questions whatsoever about any part of the lab, whether you're present or not, you're more than welcome to ask me. And in fact, because the deadlines are midnight Thursday for both labs, actually, this is quite intentional. You have time to ask me about that assignment in the next week's lab as well. So as with the lectures, we'll have time at the beginning of lab. If anyone is like, so I just don't get like chi-squared, I'm not going to say, oh, tough shit. I'll actually be able to go through any problems that you have in that first 10, 15 minutes as necessary. So don't panic too much if there's something you don't understand. Just ask questions either like privately by email. We can Zoom if necessary, or just wait until the following week's lab and we'll catch up on any issues or questions. There may be like odd things that don't work quite right, but I've done all of these labs online over the summer. So hopefully all the kinks have been, ironed out by now anyway. But as the deadline issue with labs 1 and 2 showed, I'm not immune from making mistakes, particularly if I haven't had enough coffee. This is my second cup, so hopefully I should be okay. Anybody have any more questions about the general format of the labs, just in general, the whole weirdness of alternate weeks type stuff? It's good coffee. This is actually my wife's coffee. She's out. So I'm drinking her coffee for her. There will not be pre-lab quizzes. So all of the assignment. So essentially, let me see if I can. Let's see. I'm just going to share. Dang it, where's it gone? The lab handout for today. Here we go. Let's get chat back because there was a question just as I was doing that. Question from Mackenzie. Labs are online or on paper? They are all online. So there is nothing that you need to give me in paper form. Partly that's to make sure it's even for everybody, and partly as a safety precaution. Seems a little bit maybe excessive, but given we're going to all this effort to reduce the risk of exposure to the virus to everybody, it makes sense to limit that wherever possible, and online only assignments are part of that. However, what you will find is that the assignments generally map to the lab handout questions, right? So you have, this is lab handout. I always read them before lab just so I kind of get up to speed exactly on what we're going to be doing. Essentially, they're going to be, and this is the same for most of the lab handouts, not all of them, but most of them. The questions that you find in the lab handout are going to be generally replicated in the assignment online. So it's very much as if you are filling out your handout and handing it back to me. You're going to be doing the assignment and it's going to get graded quite often by me personally, right? They're not all automatic gradings. So for example, online, I don't actually, not too worried about how tall you are. But essentially what I've done is I've given you a series of heights, right? Different height vectors. Vectors are the collection of data, essentially. And I'll be asking you to calculate sample mean of one and then the other, median, deviation, etc. So all of those questions will be found more or less in the assignment. You know, sometimes there'll be stuff that isn't in the online assignment. For example, part 2 and 3 are there for your benefit. They're not in the online assignment. However, you will be expected to draw a graph either by hand. I'll get to you in a second, Oliver Toby. Or in Excel. I'd actually recommend Excel because it's a much easier thing to use at the end of the day. And you will upload that for me into the assignment on Blackboard, right? And you'll be doing a few of these over the course of this semester, where essentially you're going to be doing something offline, you know, annotating a picture or drawing a graph, and then you need to upload that into Blackboard and I will grade that, right? So again, it's exactly the same as if you were drawing this by hand on your lab handout and handing it back in. You're going to be drawing it and then you're going to be uploading it to Blackboard and I'll see it there. So as long as they're legible, I can read it, you can do this by hand. You can, and what most students have done is they just take a picture with, well, that's now the motion, right? Because everybody uses their phones. You can take a picture of your drawing and then upload that picture to Blackboard. Or if you use Excel, you can upload the worksheet or whatever you wish, right? As long as it's in a form that I can see. So no funky Apple formats that I can't read. And I can actually understand what you've done, right? Labeling axes, data points, etc., right? So that's going to be the general format of these labs. Oliver Toby, the answer is yes. Although it's not going to be up until the afternoon because when I'm actually teaching lab in person, I'm not going to be able to get to that until probably that afternoon, maybe before I go home, maybe after I get home. But I will send an email out once I've done so. But the Zoom sessions will be uploaded the same day of the lab, sometime during that day. Erica, yes. So all of you will be doing all of your assignments on Blackboard or by email. So the two assignments that are not on Blackboard are the Excel graph that you'll be drawing from your fly data and the mini lab report, which will be obviously expansion or exposition upon that or those data. Those will be sent to me by email with your name in the file. just because that makes my life easier. Everything else will be online on Blackboard, whether you're in person or not. Obviously, if you're in person, you can do that while you're actually in lab if you wish, but you don't have to. You have until the following week to submit that. Cool questions. All right. Anybody got? Oh, correct. So the list that I'll be sending that list out again, obviously, because weirdness happened with Blackboard and my original email didn't go out. But everybody that's in the list starting 31st of August goes in next week, and 2 weeks after that, and 2 weeks after that, and 2 weeks after that. And everybody's who's in the second half of the list goes in week starting the 7th, I think. Yeah, I think that's right. For Zulika, good question. We'll be going through that when we go through the handout. I'll get through that in a bit. Simple answer is you can either do it by hand using the equations, or you can use Excel. I don't mind, but we'll go through that when we actually go through the lab handout, through the lab itself. Any other questions about just like the general course as a whole? I know this is super weird and no one's actually done it like this before, so hopefully we'll be able to wrinkle out any issues as we go along. That's the plan anyway. But if you do have any questions, ask away as always. Okay, so what I have to do first is the usual thing. You've probably seen this 101 times. Essentially, we need to go. I'm not going to go through the lab safety presentation in any particular detail because I'm sure you've seen that a whole bunch already. One thing that I will draw your attention to, I don't know where Marie got all these pictures from. It's like a fashion lineup. But yeah, you've seen all that already, I imagine. I don't know who that is who got picked on. I wonder if that's Issa, who's our chemistry lab manager. But anyway, the main thing that I'd like you to look through, and you can do this on your own time, is the beginning of this safety briefing, which is very much COVID specific, right? So this isn't what you may not have seen this before because obviously, we didn't have labs in the era of COVID before, at least not starting them. So there's a whole bunch here about using masks, the provision of masks, cleaning your workspaces, cleaning what you work with. I'm pretty sure we're going to have hand sanitizer. If we don't, obviously we have hand soap in the lab and you're able to wash your hands. Please use the same common sense that you would if you were going out to Walmart or going out in public or whatever, in terms of protecting yourself and others. And so that's just general common sense. Don't group together in groups. We're going to try and keep you apart as best as possible. And the reduced numbers are part of that, but also we're going to try and space the equipment out further so that you don't have to get close to each other. You're going to have to wear a mask during lab. We have gloves. Sorry, I got a real itchy nose. We have gloves that you can wear should you wish to do so, even if the protocol doesn't call for wearing gloves. If you want to do that as part of your own personal protection equipment. There's also something about bands. You probably know more about this than I do, like some special secret Wheatie box code or something that you have to have the right band to show that you're okay that particular day. And just use general common sense, right? So all of the lab equipment is going to be cleaned before you use it and after you use it. But if you wish to clean it yourself for touch points and things like that, then please do that too. So there's obviously a minimum that we're going to require from you to protect others, but if you wish to do extra to protect yourself further, then you're more than welcome to do that too. Get off it. The rest of this is general common sense stuff which you've seen over and over again, right? Be aware if people are around you. Dress appropriately to protect yourself and others. If there are any issues, tell me immediately. Don't try and fix them yourself because I don't get hazard pay or anything, but if I hurt myself in something, that's my own bloody fault. If you hurt yourself doing something, that's also my fault as well. So I'd rather it be me than be you for a lot of different reasons. And that's generally it. If there's anything in specific in that particular lab, then I'll tell you at the beginning of that lab to, draw it to your attention. Does anybody have any, come on. Anybody have any questions about, I'm gonna have to stop this so I can see the chat. About the lab safety or precautions for COVID that will be in place. Obviously, if you do have questions that come to you later, most likely as we're actually doing stuff, then please do ask at the time. But the general aim is to, good question, Haley. General aim is to make sure that you are safe as it is possible to make you while still being able to attend labs. Right, that's the general aim. Question from Haley about lab coats. Yes, you most certainly do need lab coats. Every single lab that you're there. If you want on Zoom, when you watch me, you can also wear a lab coat if make you feel like you're in the lab or something. But you only need to wear them when you're actually in the lab for definite. But yeah, you will need your own lab coats. I'm actually not very sure about what we're doing about if you don't have a lab coat, because we used to just rent them out, same in Gen Biolab, for a couple of bucks a lab. But I don't know if we're going to be able to do that or not currently. I'll ask the lab manager to see what she says. Mackenzie, we don't need safety goggles because we're not going to be doing anything that would require them in this lab. So no, you don't know. If you want to wear them as part of your own protection against COVID, then you're more than welcome to do so, but you're not required to wear them. Oh, that was a lovely cup of coffee. All right, so what you also need to do, I'm going to find it wherever it's gone. Here we go. Is fill out this form and upload it to that little mini ungraded assignment on Blackboard in the Lab 1 page, right? So you need to fill all this out and you need to sign it. Now, this has caused problems for people that when they save it, it doesn't show their signature, right? I don't know exactly why. My guess would be that that's an Adobe Acrobat specific deal, right? So I don't use Adobe Acrobat for various reasons. But I do know that these fillable forms are generally specific to the program that's used to make them, which I'm pretty sure in this case is Adobe. So for anybody that has had problems getting their signature to stick, right, in the PDF, safety sheet PDF that they fill out, can you please try using Adobe Acrobat? Even if you uninstall it after that, I don't really care. But try using that and see if that works. If it doesn't, let me know. Or you can just send it to me personally with your name in the file. And I'll essentially, what I need to do is kind of collate all of these together and then send them to the lab manager for her own files. Because I've gotten emails from a couple of people already about issues that they had signing the forms. Mackenzie asked a question about turning in without the signature. That's something I don't know. So this is essentially a legal document. So if it's not signed, I'm not sure if it stands as a legal document. So I will ask the lab manager, Marie, her opinion on that, because I don't want to give you the wrong information. And in the meantime, if you could try using Adobe Acrobat and see if that fixes a problem, that would be a good idea. And yes, Zuleika, coffee always, it's like French fries, always taste better when they're not your own. Definitely. I'm not going to tell my wife that I just drank her coffee because she gave me a load of grief about it the last time I did that. Oh, let's get that off. I think that's it. So if you go, hello, love, to the lab page on Blackboard. Oh, that's a very good point, Gabriella. I'll just make sure I don't tell her my YouTube handle so she can't find out the dastardly deed on her own. Anyway, so this is the assignment for the form. I don't see any reason why you can't do that, Gabriella. She was asking why you can't just print it out, physically assign it, and then upload the pictures. That will work too. Yeah, try that. That's an easy way around it, to be honest. Oluwatobi, bring stuff to lab. All you need to bring is you, your lab coat, and if you wish, your computer or device to do the online assignment, or if not, then the lab handout, right? I will not be providing lab handouts for you. So if you like doing everything online, then just bring your laptop or your tablet or whatever. If you prefer paper versions for doing stuff, then you need to print out your lab handout before lab to bring that with you. Oh, very pretty. I've just got a haircut. Okay. Any other questions before we get on to the lab itself? All right, well, as I said, if they do occur to you, then just jump. Oh, crab sticks. Yeah, got my headphone cable caught by my foot. There we go. All right, so what we're doing today is kind of a basic exercise in quantitative skills. And so what we mean by that is that it's easy to collect qualitative data, right? You know, did that meal taste nice? Or was that a big lizard or was it a small lizard, right? So that's kind of qualitative data, right? So, what could be thought of as opinions? Not necessarily, but that's kind of the general rule of thumb. But as biologists and scientists in general, we care about quantitative data, stuff that you can actually analyze and to give you answers, right? And so really that's what this lab is about, is just giving you an overview of the, oops, I better go out of this and back into, where are we at? Here we go. Back into this. That's what this lab is about. And so we're using height because it's a fairly easy metric to measure, right? Type of thing that you wish to measure. It's also a continuous measure, right? So How you deal with continuous and discrete data are quite different. So discrete data would be male or female, for example. If you're going out into the hill country and you want to look at sex ratios of lizards, for example, crickets or something, then sex is obviously, in at least the context we're using it in, a discrete variable, right? I don't know, kind of think of other ones. I don't tend to do this kind of data collection, so it's hard for me to think of examples. Um, does this lizard have a tail or has it lost its tail? If it's lost its tail, has it started regrowing? That kind of stuff, right? Whereas continuous data is where there are no steps, right? You can have, and a good example here would be how long is the regrown tail, right? Which is essentially a measure of how long ago did that lizard lose its tail, because someone was trying to eat it, most likely, or something. And so you would measure that, right? You'd use a ruler or tape measure or caliper or whatever, and that would give you a range of data or data points, uh, depending on what population you sampled. And we're going to be using height because height is just an easy one to measure. Zuleika, having a lab coat is not optional if you're in lab. You have to have a lab coat, otherwise you will not be able to take part in the lab, right? So it is absolutely, totally compulsory. The question that I don't know is what happens if you happen to forget it? What can we do in that case? And I need to check with Marie. Actually, let me make a note on my hand. We may have some disposable ones. I'm not sure. I think you can buy it in the bookstore. Don't think I've ever actually been in the bookstore, so I don't know. But you can certainly buy them on Amazon. There you go, for example. And I imagine the prices would be comparable, if not better, than in the bookstore. But again, I don't know. I'll leave you to, figure that out. But yes, for the first time not sure about next week. We might, maybe if you struggle to get a lab coat by then, we may be able to excuse that. I'm not sure, but certainly after that, yes, you will most definitely need a lab coat. Otherwise, you won't be able to attend. For the, I think Gabriella said she bought hers on Amazon. Gabriella, if you could post that link up that you used to the chat, then I'll upload the chat to Blackboard after lab, and so that'll be there for everybody. Awesome sauce. Anyway, so essentially one of the big problems that we have as biologists is that we typically can only measure a subsection, a small part of a population, because for various reasons. One, it may be impossible to measure the entire population because you can't find it, right? It's not physically present when you're doing the measurement process. It could be that the population size is too large for it to be feasible to actually measure all of the population. All right, if we were doing height, for example, at the very minimum, you would want to measure the height of everybody on campus, right? That would be your minimum population. Really, I mean, you probably want to count everybody in San Antonio, right? And that's just not feasible. It's not manageable doing that. So as biologists, we inherently acknowledge the fact that we're going to be dealing with a small subset of the population, right? So that's one issue. The second issue is that biology is inherently noisy, right? That may be, I mean, even especially for continuous traits such as height or tail length or whatever, but even for discrete characteristics, right? If you measure sex ratio, if you measure sex ratio in uh, in the lab, for example, you're most likely going to get a slightly different answer, or you certainly will actually, because typically speaking, it's skewed towards females in biological sciences at university. Um, Not always, but typically it is. But even if it wasn't, there will be an inherent noise, right? Which is both because biology is noisy and also because you're measuring again, a subset of that population, right? So if you measure, say for example, 12 students that come into lab for their sex, well, not measure it, you just need to look uh, at their faces, right? Just for get any kind of ideas. Um, but even if you do that, it's unlikely that you'll get 6 and 6, male and female, right? And without any external forces acting on it, sex ratio should be 50-50, right? So you may get 7 and 5, or 5 and 7, or maybe even 4 and 8, right? So if you calculate sex ratio from those numbers, then inherently you're going to get a skewed result, right? So what we want to know is how accurate, how far off of the population is that particular sample, and is it differing in ways that are meaningful, right? So for example, I said about sex ratio in biology students, right? It may be that there is some other explanation other than sampling bias for why there are more females taking biology than males. I don't know. Is it something that is more appealing to those, or less appealing to males? I don't know. And so those kinds of questions lead to interesting kind of uh, further questions. And that's really what this kind of lab is about, right? Is how do we go from the sample that we measure to extrapolating that to a population level on one hand, and on the other hand, trying to figure out, are the differences that we observe, are they meaningful, right? So if males are different in height to females, is there actually a reason for that? Or is it because it's just as a result of our sampling, right? And so that's essentially what we're going to be doing today. And so typically whenever we do a sampling, whenever we measure something, whatever that might be, we want to kind of take all those numbers down and turn it into something that we can handle. Because if you were to look at, uh, you know, a huge range of data, it's really, really difficult to figure out what's going on. Let me see. Oh yeah, I've got a great example for you here. This is something that I actually haven't gotten around to analyzing, just because it's like, oh man, this is tricky. And I haven't had time um, this summer, basically from teaching. Right, so this is a qPCR data analysis, a quantitative PCR data analysis, looking at gene expression of these particular genes here, right? They've got different names and stuff. So if one were to look at this, and so this is essentially looking at changes in gene expression expressed as a ratio or a fold change, right? So more expressed or less expressed, right? And just looking at this data is like, what's going on, right? So it's really hard to figure out exactly what the picture is, right? So the very first thing one needs to do, right, and there's just tons of numbers in here, oh, loads of numbers, is to reduce those numbers down to a form that is more inherently accessible to us, right? Our brains work in particular ways, and we're pretty crappy at looking at large number of data points and inferring meaning from those, right? It's very easy to get confused by the noise. And so one of the first things that you do is you calculate a mean, right? And that's what I need to do for those data. Just haven't got around to it yet. And then the problem is though, all right, and so this is something that, I deal with constantly in the kind of data that I collect for my own research. As soon as you take all of those data points and you convert that into a single point, which is the mean or median, if that's more appropriate, you lose all of that other information, right? So this graph here is a great example of that, right? So these are all normally distributed data points, right? Presumably continuous data for whatever that variable is, right? And they all have exactly the same mean, right? However, if you expressed those three different samples just by their means, they all look the same. If you look at the data expressed here in graph format, they clearly don't look the same, right? They are different, right? And so we need some means of adding that information back, right? And what we do is we calculate the variance. So essentially, as you can see here, our mean is right straight down the middle, right? In the middle of this graph, the center of the bell curve, essentially. What you can see is that this sample, whatever this black line represents, it doesn't differ very much around. I think these are actually standard deviations, 1, 2, and 3 standard deviations, but that doesn't matter. You can see that the data do not vary hugely around the mean, right? So almost all of the data is very close to the mean, right? So one would say that this sample has low variance, or if you were to calculate it, it would have a small standard deviation, right? Which is the figure that we actually use. Pardon me. Now, if you look at the blue sample and the green samples, right? They too have the same mean as the black one, but they will have differing degrees of variance. With green having greater variance, there are more data points which are further from the mean than there are for the other two. And the blue is somewhat in between. So as soon as you calculate the mean, you lose all of that information, right? It's gone. But that information is important because it tells us a lot about what that sample looks like, and from that sample, presumably that population. So we need to basically add that back. And to do that, we'll be calculating the standard deviation, right? So first thing that you're going to do is I'm going to give you the vectors for height. So in each question where I'm going to ask you mean, median, and so on, I give you the data there, so you don't have to keep referring back to prior questions, right? So you're going to have those data and you're going to work from them. They're also going to be obviously quite precise answers from those data, right? So when you present your mean, for example, so your mean equals, what would it be? Value to 1 decimal place. So for example, this is often a question that I get. E.g., if the mean is, you calculate it as 164.58, oops, there's supposed to be a 0.58, for example. You'll put that into the assignment as 164.6 centimeters, right? So I don't need reams and reams and reams and reams of decimal places because that's typically not all that informative. So you need to round it up to 1 decimal place, but obviously if this was 164.48, that would be 164.5, for example, right? That's what we mean by rounding up. Okay, so you can calculate this by hand. Essentially, what you do is you add all of those data points together and divide them by the mean, uh, by the sample size. Sorry, that's what the n is, right? So if you have 7 data points, then it's that total divided by 7. What you can also do, and let's see. I'm not going to do anything in here because it would just take me way too long. I have to actually even think about what I'm going to be doing. Can I? Here we go. All right, so I do everything in Excel because it's easier. As long as you understand the principles that you're going to be working with, right? That's important because otherwise you can make mistakes through ignorance. So let's say we have right, these are all our data points. Can you all see what I'm doing? Hopefully you can. My chat, come on, dang it. No, that doesn't work. It's really hard for me to get chat back for some reason. I really don't understand why. Oh, now it's back again. Who knows what? Okay, so the way that I do it, typically speaking, is I use the functions in Excel. So if you want to use Excel, and you don't have to, you can do all of this by hand quite successfully. And it wouldn't be a bad idea to try doing a little bit of that anyway, right? If you ever want to use a function in Excel, hit equals. And as soon as you do so, you will see in this little box up here, which is all your functions, it typically suggests one based on what you used last normally. And if you want, you can click on that down arrow and select something different, right? So I want to do average, and that often selects makes a guess essentially on the selection. I don't want A1 to 6 because A6 is empty. So I click on, let's just go back to that actually. Click on this button, dunk, and then select those boxes and hit return. Or if you you can do it by hand, right? So you just go in here and then type, 5 instead of 6. Sometimes it's actually a good idea, just so you don't get lost, giving it a little label, right? And so this value here is the mean or the average of these 4. So essentially what this is doing is adding these 5, it's 5 different data points together and then divided by 5, right? And you can double check that by hand if you wish. Right, so that's the first thing that you need to calculate because that gives you an idea about more or less what's in the middle. Now, because you'll make an assumption that these data are normal, i.e. they are bell curved, right? There may be situations in which case that's not true, right? Income actually is a great example of that. So if you look at income in the US, for example, the mean, would be, I don't know, I think it's like $60,000, like mean family income or something. But if you look at, but that's actually essentially a lot of people not earning very much and a few people earning an enormous amount, right? So that the data is very heavily skewed towards lower incomes in terms of the number of people earning a certain amount, right? So another way of looking at this to deal with that skew or skewedness is to look at the median. And really what the median is doing is, actually we can do this by hand. 165, it's laying out these data points in order. So 165, 172, hopefully I don't screw this up. 187, 188, and 191. Done. So the median is quite simply the middle one in the line, right? So you put these out from smallest to biggest or biggest to smallest or whatever, right? Doesn't really matter. One extreme to the other, and the one that's in the middle is the median, right? Or it could be, if there's an even number, it's going to be essentially an average of the two that are in the middle to split the difference. Or you could be, I don't know, I was going to say lazy, smart maybe, kind of could be both. And use the median function, right? So again, equals, go up to functions, and then select the data that you want. Ah, it worked. Awesome, right? So I didn't make a mistake, which is always nice. It doesn't hurt to double check sometimes, right? So essentially, those two are the same, right? Which they should be if you get this right. Okay, anybody have any questions so far? See if I can get, come on, God dang it. It's intensely frustrating not being able to see the chat. Let's see. No, it's Zoom is flaky as when it comes to stuff like that. I don't really understand why. Oh, well. If you got questions, oh, now it's back again. Don't know what that does. Oh, now it says my screen sharing is paused. Great. All right. So if you've got questions, you're just going to have to unmute your microphone, go, and then I'll figure out a way of seeing the chat. I'll see what your question is. So going back to the handout, wherever that's gone. Here we go. That's essentially explaining what these are, right? So, you know, that's the first part. And these will be the first questions. Where are we at, right? Essentially from 3 onwards, right? That's what I'll be asking you to do in the online assignment on Blackboard. What I want you to do next is start calculating these following things. We want to have sample variance, right? And so essentially, sample variance is how the sum of how much these individual data points are different from the mean. So if we go back to this graph here, if we calculated that for all the points along this line for the black line, it would be this point here minus the mean, 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 so on and so forth, right? For all of the points that are on that line. And then we do a little bit of jiggery-pokery with them, where essentially we square that difference, right? The reason why we square that difference is because if you subtract essentially half of your values, if you do this right, should be less than the mean, right? So if you were to do this without squaring them, you would get negative values. When you add negative and positive values, they cancel each other out, right? So you wouldn't actually see the true variance. So for every single data point, you would subtract the mean from the data point and then square it, right? And so you can do that just by hand, right? So essentially it would be, or you could do it in Excel if you want. It would be this minus this, right? That's a positive number, right? So what we really need to do is put that in brackets, right? So that kind of keeps it in there. And crap, what's a square? Square, square, square. I think that's Hmm. Hang on. Let's go back. Yeah, I can't do the square root. Okay, so let's do it the clunky ass way, right? Which is this minus this times, which is the asterisk, and then the same thing again. Essentially, that's squared. It's kind of a clunky way of doing it. They all work. And so you can do that for all of those, right? And if you kind of drag that down, it'll look all kinds of weird because you're starting to use the median. Right, so you'd need to change that, right? So you can do that for all of those, right? Or, and actually you'll need to do that because I think that's a question. I'd have to double check. Let me have a little look. Oh, it's always, because the lab handouts differ ever so slightly from the assignments. I want to make sure that I'm actually asking the same things. All right, I'm actually asking you for variance. Okay, so let's make sure that we do that. Right, so you can do this all by hand. You can do it in Excel, right, which is a little bit tedious, but it's not impossible to do. Right, so essentially what I'm doing is calculating the sum of the differences. We're not quite at variation yet because there's a little bit more to it than that. So, oops, forgot that one. Right, so let's just double check. Those should all read B7 at the end. Okay. So these are all the individual variances for each point. Now, if we want variance, I use Excel a lot, which is if I go too fast, please say, whoa, horsey, and get me to back up a little bit. Again, this is all laid out quite nicely in the handout, so you can actually do this by hand. I'll show you I think I can work out variance in Excel. I tend not to because I use standard deviation instead, and we'll get to that. So essentially, variance is all of these added together, divided by sample size minus 1, right? And sample size is 5, obviously. Let's do n here. You can also do that if you're like really lazy and do a count.
Are you supposed to be showing us Excel?
I am I not?
No.
Oh, crap sticks. I'm sorry, I'm probably in the, here we go. So I was trying, let's just share the screen, right? And because the problem is I definitely can't get the chat up in when I share my screen for, I think that, oh, there you go, I can, it just appeared now. I don't know why. This is a bit of a frustration, but we're just gonna have to deal with this. Weird that that didn't work. Anyway, that usually works, just gonna do it by hand. Haha. Okay, so variance is the sum of all of these, right? But we want to make that one calculation. So we're going to, let's see if this works. Bugger off. Let's go back. And, nah, that's not gonna work. I'm probably trying to do, I'm probably confusing you more than I'm helping you, aren't I? All right, so that's all of those individual sample minus, or data point minus the mean squareds added together, right? So if you go back to Where we're at, what we're looking at here is this top part of the line, right? So this is the sum of the sample minus, or the data point minus the mean squared. Does that make sense? Hopefully it does. So the variance is that divided by n, which is this number, minus 1. And so that's a pretty big number, but that's what the thing tells us to do. Let's see if that's correct. Let's see if we can do this. I think we can. Var S, I think, is var sample. Oh, no, that didn't work. No, that's not right either. I don't understand that function. I never used it anyway. So that's the variance, right? That's the next thing you need to add. But the problem with variance is essentially this is going to be centimeters squared, right? Because you're squaring these numbers, and those units carry over, right? And obviously our height is not a function of area, right? It's a linear measurement. And so you don't want that to happen. So what we actually want to do is standard deviation, right? Which I always write as SD. And all standard deviation is the square root of variance, right? And square rooting basically gets rid of the square-ish bit, right? Gets rid of that 2 after it, right? And so you can do that by hand on your calculator. You can do that. I do know how to do this one. This is SQRT. Yes, that's what I want. I want that, right? Or you could actually use the standard deviation function, which is what I do. So that's stdev here, and it comes out as something completely different. I think I did that one right. This happened last time. I screwed it up somewhere. I can't remember what I did. Oh, numbnuts. It's this one. Oh, come on, back again. Obviously, that's not going to be right. There you go. Whew. Phew. Right, that would have been really embarrassing. And now our unit is in centimeters, right? So we're back to what we need. Okay, so essentially what we're saying is that our mean equals, what is it, 180.6 plus, I'm not sure if this will actually let me write this in Excel or not, 11.4 centimeters. No, it does, right? So that information now tells us, okay, we've got these 5 individuals. Our mean is more or less in the middle, give or take. But because we've got some people that are really quite tall and some people who are quite short, essentially we have a pretty wide, distribution of data points. Now, what will happen? Oh, actually, let me ask you. So what do you think would happen if this sample was representative of the population, and now you measured 100 people instead of 5? Now let's say that the mean that you calculated is representative of the population. What would you expect to happen to the standard deviation? This is a question to the class. So this is a pretty small sample of 5. Right, so the standard deviation gets smaller the larger your sample size. And so essentially what that is telling you is that you are more and more confident that your mean falls within 2 extremes, right? So measuring 5 people, we think a mean height is 100, basically between, what's that, 169 and 191 centimeters, right? Now, when we look at a larger sample size, it might be 100, our mean is somewhere between, with a degree of confidence, let's say 175 and 185, right? So the bigger the sample size, the more confident you are that your mean is actually the same as the population mean. Your sample mean is the same as the population mean, right? Now, another way of dealing with that is what's called the standard error of the mean, SEM. And so essentially what that is the standard deviation divided by the square root of the sample size. Where are we at? Okay, so essentially this is for sample variance. This is for standard deviation, which is a square root of the sample variance. Now, if we, let's say this is particularly good if you're doing repeated measures, right? So we measure 5 people from the population over here, we measure 5 people over here, we measure 5 people over here, right? So how do we express our confidence? Essentially, every time we repeat this with different people, we should be more confident that the mean that we get is truly representative of the population mean, right? And so to be able to express that, what we do is we would take the average mean of those 3, and then we'd calculate the standard error of the mean. And what this does is it basically gives is able to correct for sample size, right? So where we go back here, that would be standard deviation would be that divided by the square root SQ, where we at? Here we are, of the mean. Oops. Yeah, that looks right. No, that's not right. If in doubt, put it in parentheses. No, that's not right either. Dang, I'm gonna have to use an actual calculator here. This is what I normally do. What's 5? We need scientific, please. 5 square root. Yeah, 2.24. I don't know why that didn't work. Anyway. Again, I always just work this out by hand. Right, so, oops, crab sticks. Let's get rid of that. Oh, I'm sorry. So the SEM is essentially makes the standard deviation smaller depending on the size of the sample. So the standard error of the mean is the standard deviation divided by the square root of the sample size, which is going to be n, right? So that's going to be square root of n, which is 2.24 in this case. Am I just doing something completely? Oh, that is right. I'm sorry, I'm a little bit slow today. Yeah, I kept looking at the n. It's like, why is it the same number as the n? Obviously, because I'm looking at the wrong thing, right? So now what you would do And this is right. Now you would express this as the mean equals this plus or minus 5.09 centimeters instead. And this is standard error of the mean. Oh, so I was actually doing it right. Yeah, wonders of things. So essentially, you can see that we now have greater confidence that the mean is within a smaller range, right? So if one were to count 20, right? So for example, let's see if, what's the square root of 20? Let's see if I do this right. So if the n equals 5, the standard error of the mean would be 5.09. Well, that's actually 5.1. Oops. If I'm rounding up properly. Now, if the SEM n equals, let's say, 20, oops, great. The square root of 20 is going to be larger than 5. So you're going to be dividing by a larger denominator. What's the other one underneath? Denominator, I think. So your standard error of the mean should be smaller. Let's see if that's true. Okay, so we have our standard deviation divided by square root of actually, let's do this by hand. I'm not entirely sure how to do this without confusing you or me further. Right, so the square root of 20 is 4.47. And so now you can see that the standard error of the mean for a larger sample size is going to be smaller, right? So as your sample size gets larger, there are limits to how effective this can be, right? It kind of starts tailing off after a certain point. But as your sample size gets larger, your confidence in the accuracy of that mean gets greater, right? So the range in which that mean will exist gets smaller, right? And so really what this is trying to do is to take into account how big is your sample size. And you've always been told, you know, bigger sample sizes are better. Well, this is why, right? Because the larger your sample size, the more confident you are in your mean representing the population mean, right? The sample mean being the same as the population mean. And it'll never be exactly the same, right? That's, unless you actually measure the whole population, that's not statistically possible. But you will be able to get closer and closer to that and have greater confidence that you're actually truly measuring the population mean, or looking at what's going on in the population rather than just what's going on in your sample. And so what you need to do for your last assignment for today, and again, if all that Excel stuff just sounds like complete gibberish to you, that's fine. You can watch it over on again on the Zoom if that helps. And if it doesn't, you can do this all by hand, right? So I think there are 7 data points in the vectors that I give you, I think, something like that. So you can actually just work out individually what those, what your sample variance is, what your standard deviation is, simply by following the handout, right? All that I was trying to do by showing you the Excel, and I realized I do this a lot, so it's quite easy for me to just chunter on, and because I know what I'm doing. There's a risk that I lose you, right? And that's not intentional. I'll actually save this and upload it to the lab afterwards because it was maybe useful to have like the spreadsheet to be able to compare the video to. But if you were to want to use Excel, that's actually in many ways a more straightforward way of doing these kinds of calculations, right? And obviously when you have 7 data points, it doesn't take that long. But if you wanted to calculate standard deviation of, a couple of hundred, it would take you ages, right? With the, associated risk of errors. So if you want to use Excel, that's also fine too. Please do that. Now, the last point to talk about is I actually want you to draw a graph, right? So I want you to take the means, right? So I've just calculated for one vector. You're going to have 2. You're going to have male and female vectors. I want you to calculate the mean plus and minus the standard error of the mean. Do I ask standard error or standard deviation? Oh yeah, standard error of the mean. Yeah, there you go. So you do need to do the standard error of mean. And I want you to do a dot or a line, I don't really mind, here. And I want you to draw error bars, which is essentially you draw up a bar from a line, right? So, aha. Well, this is blue. That's not ideal, but color is largely irrelevant. Oh, my dog's just wandered off. She's scared that I'm going to do something to her. Well, color is relevant because this one sucks. Where are all my different colors gone? There we go. Expo for the win. Right, so this is going to be a graph. You could even draw on a whiteboard like this if you wish, as long as I can see what you're doing of male, female, whichever way around you wish. You're going to need to, this is going to be height, hopefully written a little bit more legibly than that. Units, centimeter, right, which are always in parentheses. And I don't know, let's say you don't have to go down to 0, right? Because no one in this class, hopefully, is 0 centimeters. So for your males, I don't know where it would be. Let's say here. This would be your mean. You can do it as a star if you wish. I don't really mind. Typically, it's done as a line because that's going to be your mean for that sample. And then with your calculated SEM, which is going to be your mean, let's say plus, let's say 5, right? So that's going to get you to here. You're going to draw a little bar across the top of that, and then you're going to do the same for the minus. And that is your mean plus and minus the standard error of the mean. And so essentially what you're saying is that you're pretty confident that your mean exists within this range, right? I'm just going to rub that off because you don't need to put that on the graph. And typically, but not always, females are a little bit shorter than males. And then you'll do the same there. I'm drawing the SEMs as the same. They may not be. I'm just doing it because I don't have any other data to say otherwise, right? And that is the kind of thing that you need to do and upload that to your Blackboard assignment. And that's pretty much it. There is other stuff in the handout, which is about scientific notation and also dilutions. That's useful stuff to go over, particularly the dilutions, because we'll actually be using that later on in one of the labs to calculate dilution of a DNA or PCR. I can't remember which. DNA, I think. And that's pretty much it. So I'm going to save this Excel spreadsheet to the lab page, wherever that is. Oh, dang it. Obviously haven't used an Excel spreadsheet in, have a little look at my hard drive here. Isn't that fun? Let's actually put that in lab 1. Oh, look at that. I actually did that. There you go. So I'll post that to Blackboard in a little bit. Maybe I might have some lunch first, I'm not sure, but that'll be up and around within the hour for you, as will the chat. The Zoom session will be however long it takes Zoom to convert it and then YouTube to upload it, which will probably be about the same sort of time. Does anybody have any questions?
I was going to ask if you can show how you got the 2.55.
Oh, this was really just an illustration of how SEM varies based on sample size. So the correct SEM for this sample size is 5.0 or 5.1, right? Because that is the standard deviation, which is this number here, or this number, whatever you want to do. It's that number divided by the square root of 5, which is 2. No, it's like, I can't
Yeah,
remember
it's
what it is actually.
2.23.
Yeah, that one. Now, if you have a larger sample size, this is not the correct SEM for this data set because this data set only has 5 sample points. But let's say you did get that same mean from a larger, 4 times larger data set, then your SEM for that would be smaller simply because your denominator in the SEM calculation is going to be larger, because the square root of 20 is going to be larger than the square root of 5. That's really it.
Okay.
And so really, this is the correct one, right? This would be, if these were the data points that you were going to be working with for the assignment, this is the SEM value that you would plot on the graph. This one is purely to just show you that the SEM will decrease essentially the larger the sample size. So if you did SEM n equals 100, because that's easy to calculate, right? That's going to be the standard deviation divided by 10, right? So again, it gets smaller still,
Oh, okay.
right? Do 1,000, it'll be even smaller, right? At some point, it'll level off, right? You won't get any appreciable gains in smaller SEMs. But certainly for the kind of sample sizes that we work with, increasing the sample size increases the confidence that you have that your mean is correct or represents population mean, because your window gets smaller, right? So essentially the SEM is a measure of confidence based on sample size.
Okay.
Bigger the
And
sample
can you
size.
click?
Say again?
Oh, I was gonna ask if you can click on the mean and median so I can see what functions you use.
So I'll also
Oh,
upload
average.
this to Blackboard so you can kind of wander around this yourself.
Okay.
So for some reason, Excel calls as mean average. Maybe that's because that's the general colloquial term for mean. The median is actually just called median. So what
Okay.
I tend to do, and the same for standard deviation is called STDEV. But if you put the equal sign in, it will automatically bring up this. And if you want something that isn't there, click on more functions and square root, for example, and that will bring up the function that you need to use to calculate that particular thing.
Okay. And with the sum, did you do the sum with all of the numbers, like, or just B1 through B5?
B1 through B5.
Oh
So,
no.
okay, so this is, be careful here because
Yeah.
what I'm doing is I'm actually calculating the variance. And so because the doing it in Excel would result in a really tricky calculate equation to write, which I might screw up because I don't do it very often. What I did first was I calculated the square of the difference between the data point and the sample mean, right? So that's data point 1 minus mean squared. It's actually easier to do this by hand, to be honest. Data point 2, right, minus the mean squared. And then the sample variance is all of those added together. That's what the sum is, divided by n minus 1, which is 5 minus 1. That gives you the sample variance.
Okay.
And the standard deviation is that, the square root of that, essentially. Does that make sense?
Yes. Yeah, I just didn't get to see the functions.
Oh, okay. Yeah, so have a look through this when I post it up in a little bit, and you'll be able to hopefully follow what I've done. If you don't, just ask some the questions and I'll be able to answer
Okay.
them for you.
Okay, thank you.
Illy, good question. Essentially, I give you the data points to work with. So every person in the lab has the same set of data points. So you don't need to do this yourself. I've provided the data points for you. You simply need to calculate the means, medians, and so on. All right. Well, thanks for your time, everybody. Hopefully that all made sense. If it doesn't, ask me questions by email or save them for lab next week, and I'll be able to answer them then for you. All right. Have a lovely rest of the day, everybody. I shall see you soon.